Hi everyone, I'm Noor Pratap Singh, and today I'll be talking about Alevin-fry-atac, which is a method for processing single-cell ATAC-Seq data. So, just to give a brief overview, so ATAC-Seq is a technology that enables profiling of the open chromatin regions, and this provides insights into the regulatory landscape. Now, the way it works is that we have this enzyme TNF5 transposes. This is loaded with DNA adapters, and these are added to a bunch of extracted nuclei, isolated nuclei. Now, this has a property that it simultaneously cuts the DNA and inserts these adapters into these regions of open chromatin. And now these extracted fragments can accordingly be amplified and sequenced. So, coming to single cell, there are many technologies have exist, but the protocol by 10x remains among the widely used one. Not going into the details of this protocol, I think the key thing to take away is that in addition to the fragment, we also have barcode information with us, which encodes which cell that particular fragment came from. So, in a typical uh, ATAC-Seq, single cell ATAC-Seq workflow, there are a bunch of different steps. But as in this particular framework that I'm talking about, we care about the top three steps. And again, in this entire set of steps, alignment and mapping is a key part because usually it's the slowest steps in terms of computation. So, talking about some of the existing work, like there are three main branches we can think about. The first one is the tool Cell Danger Attack. Again, this is by 10x, and what it does is it's an end-to-end pipeline, and which does apart from mapping and processing, does also other downstream steps, including peak calling and clustering and differential peak analysis. And the mapping is slightly slow for this particular tool as it uses a variation of VWMEM for mapping. Then we have ChroMap. Again, this is a fully open source method, and currently was at that time was the fastest method for doing such analysis. And in the third category, we have a set of pipelines such as Maestro, which stitch together different tools for each of these individual steps, such as like Star or Minimap 2 for mapping, and then using SAM tools or PCARD for to sort and filter the alignments. So, Alevin-fry-atac, what it takes as an input is the single cell ATAC-Seq reads and outputs a bed file that contains the cell barcode corrected, deduplicated, coordinated, sorted map reads. Now, this bed file can be passed as an input to other downstream tools, such as for peak calling, or which in conjunction later, then also be passed to Signet for clustering or other analysis of interest. So the Alevin-fry-atac framework consists of three parts: indexing, mapping, and downstream processing. Breaking each of these components. So for indexing, we use something called a PySim index, which is a modular index. And the heart of it is a De Bruijn graph. If anyone speaks Dutch, please forgive my pronunciation. So this is a commonly used data structure for representing a collection of sequences, and we use cuttlefish for generating it. So uh, talking more about De Bruijn graphs, so what we have used is an edge-centric representation. So with each node represents a k-mer, and with any two nodes that are connected by an edge having an k-1 overlap. Uh, Specifically, though, it's what we have is a colored De Bruijn graph where each node, which also encodes the information of the color or the references from which that particular node exists. And so, but for the practical purposes, what we have is something called a color compacted De Bruijn graph. And the idea again is that we want to compress these nodes into a collection of nodes into one single node called a unitic, which represents the maximum non-branching path, such that each internal node within it has only one incoming and one outgoing edge. So once we have this De Bruijn graph or compacted De Bruijn graph by Cuttlefish, what we have used is SSHash. So it's an efficient k-mer dictionary, and what we use it is to map each k-mer to a unitic Along with that, we also it gives us the we use it for encoding the information where that k-mer mapped within that unitic along with that orientation. And then the final component of this index is a dense inverted index, and this is used for mapping a unitic to the reference, encoding where exactly that unitic occurs within reference along with the relative positions and orientations. And we call this U to R. Together, K to U and U to R can be stitched together to extract where exactly k-mer maps within the reference. Coming to mapping, what we are using is called a pseudo alignment strategy, and what we have added is our virtual colors. So pseudo alignment is a lightweight mapping strategy that seeks to test the compatibility between a read and a reference without actually not caring about whether the base level coordinates of each base within a read. And in most cases, when we have small reference references like transcriptome or a metagenome, it has been found that the accuracy of pseudo alignment is comparable to alignment-based approaches. Again, there are different variations of pseudo alignment, but what we have used something called a hybrid or the threshold union approach, where we idea is that we break a given query or a read sequence into a set of k-mers, and for each k-mer, then we extract the corresponding references that they exist in. And then, depending on if a certain proportion of k-mers map to a reference within that read, then that reference is assigned to that particular query. So in this case, let's say if I have a threshold of 0.7, then for this particular example that I have, we both R1 and R2 are the plausible mapping for that query sequence. While for short reference sequences, pseudo alignment is a good strategy, but when we have large reference sequences such as the genome, and where let's say the colors would be the individual chromosomes, we observe that there is a drop in accuracy with respect to mapping. And this happens because there exists multiple mapping location for a k-mer within a single reference. And just to provide an example, let's say I have just a read with just two k-mers, let's say. And now if you look at it, in the first chromosome, this k-mer maps at 3 position, and in chromosome 2 only in 1 position. And the k-mer 2 again in chromosome 1 and chromosome 2 maps in 1 position. Doing a standard pseudo alignment, we will have it will output chromosome 1 and chromosome 2, but we also try to care about the mapping position. And the way pseudo alignment outputs a mapping position is based on the position of the leftmost k-mer. In this case, since the leftmost k-mer is mapping to 3 positions within the first reference itself, so what usually ends up taking is the first mapped position. So this is what we get as an output, and we can see that this is neither this nor this is the place where this read maps. So we have 2 false positives. At the same time, we have a false negative because we have missed this mapping position. So what we propose is doing adding something called virtual colors. And the idea is that we project these original reference sequences onto a set of virtual colors that do not necessarily reside on the index, but will represent the targets to which the queries can map. And these virtual colors effect consists of set of overlapping bins. And the idea of overlapping bins is needed is because it might happen that the entire read might not fit within this artificial bin or the virtual color that we have proposed. And so we have this set of something called overlapping bins. And the only caveat to keep in mind is that that overlapping region should be longer than the read length, so that that entire read can fit within that bin. And just to now provide this example, in this particular case, the k-mer 1 maps to that overlapping, is in that overlapping bin region. So we give it 2 colors, B2 and B3, such that when the pseudo alignment happens, we are able to correctly map the read, and the final bin ID is B3 and with the position 1, 2. And now B3 then can be mapped later to the chromosome to get the actual location. So far, the strategies that I talked about that improved the accuracy of pseudo alignment, and then in the next, what we worked on was improving the overall mapping speed. So we specifically introduced two key innovations. The first one is about using streaming queries, and the key thing to note is that the way we want to improve the mapping is that we want to minimize the uh, um, accesses to the main index for extracting that schema information. So assuming, like, say, if for a given the way we do it is that it's very likely if two schemas are at consecutive positions within a single read, then it's highly likely that they might also be on the same unitic. So assuming if I had the let's say this came a TCA, and I had the unitic information for it, and then when I'm trying to extract the Kmer position for CAT, then rather than looking at the main index, I can go back to the previous unitic, and from that I can accordingly, based on, because I have the direction information, I can accordingly extract a corresponding nucleotide and then match it with this next nucleotide on that Kmer. And if they match, then I have that mapping information. And if that's not the case, then I can go back to the main index. The second key idea that we exploit is that a lot of these attack secrets enriched at certain locations. So we had to query the main index again was because whenever we were entering a new unitic. But let's say if this is there's a commonly occurring path, then we can again also then store these starting or the terminal kmers that start at the beginning of a unitic in a cache. And then whenever we are trying to query, we will try to see if the unitic information exists. If that doesn't exist, we look the cache. And if it's not in the cache, only then we will look at the main index. And introducing these optimizations like end up like say like our mapping speed increases by 30-40%. And then we have these standard processing steps like cell barcode correction, which corrects for the observed barcodes that are not present in the whitelist file, and then sorting and deduplication, which removes the deduplicated fragments and then does coordinate sorting. Looking at some of the results, so we first evaluated this method on simulated data sets. So what we created used Mason to generate simulated reads of different lengths, and then try to look at the mapping accuracy. And what we have is that we varied the bill length that we had. And we see that there is a fall in accuracy as we keep increasing the bill length, with the sharpest fall being when that entire chromosome is used as a color or no binning. And accuracy falls somewhere between, let's say, around 94% for the bill length of 1,000 to somewhere around 84%. And then we also compared the overall mapping accuracy with alignment-based methods like chroma and bow tie. And what we observe is that, yeah, we have lower accuracy compared to them and across the read lengths, but the difference decreases as we increase the read length. Then we actually compared the results on the experimental data sets and specifically the downstream analysis, and see if the loss mappings have an impact there. And what we did was, for the different data sets, we compared the clusters that are obtained, and you use these metrics like adjusted brand index and NMI for comparing the clusters. And higher the value, higher the concordance. And what we observe is that across the data sets, there's high concordance that is observed between different methods. And in fact, on the 10K human 10K PBMC, there's high concordance between Alevin-fry-atac and Cell Ranger compared to chromap or Cell Ranger. Finally, talking about the computation speed, we look at both, compared the running time and the memory usage. And what we see that is that at lower threads, number of threads, chromap is faster. But as we keep increasing the thread count, the scalability of Alevin-fry-atac is better. In around 12 threads, it starts improving. And at around 32 threads, the speed it's almost 3 times faster. Similarly, though, consistently across the threads, the memory usage of Alevin-fry-atac is like it's 3 times smaller than ChroMap. And though I have in this table results for cell-ranger attack as well, but it's hard to directly compare because it's an end-to-end pipeline and it's not feasible to actually run or benchmark each individual step. And a thing I would like to note is that the comparisons with ChroMap were done with the version 0.2.7 for this manuscript. They recently released the version 0.3.1, and there they have reported an improved scalability. But again, I haven't had the chance to do the exact comparison with the most recent version. So to summarize, we have Alevin-fry-atac, which is a memory efficient and ultra fast tool for processing single cell ATAC data. We have proposed a novel pseudo-alignment procedure now, which we hope which can be applied to large reference sequences such as the genome. But another importantly, now this Alevin-fry-atac is integrated into the Alevin-fry ecosystem and SimpleF, and this integration creates a unified ecosystem that simplifies and improves multimodal analysis. And the goal is that if we'll also plan to add other modalities so that in one ecosystem you can do all the analysis. The workflow and the packages are available on GitHub. Yeah. And finally, I'd like to thank my advisor, Dr. Rob Patro, Julio, who's a collaborator and developer of SSH, and Jason and Jamshed are the former PhD students in the lab, and Jamshed developed Cuttlefish. Thank you. Happy to take any questions.
Thank you so much for the talk. Are there any questions? Please move to the microphones to ask questions.
Hi.
Hello, thank you for the presentation. So I have a question. When you were doing the optimization for overlapping k-mers, you looked into the unitic, but can you in this way sometimes lose some of the matches to other unitics, or do you recover them?
A k-mer actually maps uniquely to a unitic, so it's only one unitic a k-mer will map to, right? So
but the next one can have multiple matches, or no?
Oh yeah. So that's why we are storing state information. If the previous k-mer is at the boundary, then we have to query the index again. So it's only if that k-mer is within that unitic boundary, then only it will happen, right?
So I was thinking about the first optimization where you sometimes don't look into the index because you find the match in the unitic where the previous k-mer was. But
yes.
Whether sometimes it cannot happen that the next k-mer will have multiple matches and you don't find them.
But a k-mer will be uniquely mapped to a unitic. So even the next k-mer, if there's a match.
Oh, okay, I understand. Okay.
So yeah, so it will always be within that unitic. Yeah.
Okay, thank you.
I actually have a question. So you had to you're trading some accuracy for the speed, right?
Yes.
So have you tried to understand when you're like that the reduced accuracy is you have issues with the mapping on some places, whether there's any bias in it and what it means for the downstream results?
Yes. So so that's why we try to do this this clustering analysis like this ATAC-Seq. So at least there we find high concordance. So that was sort of our check in a way that it's not impacted on that part because that was the key target application. But then again, where exactly those loss states would impact is something we have not explored that yet.
Okay, thank you so much. Let's thank the speaker again.